Chapter 16 Excretory Products and Their Elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chlorine, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with special emphasis on common nitrogenous waste. Ammonia, urea, uric acid are the major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water from its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia is ammonotelism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface in fish as ammonia ions. Kidneys do not play any significant role in its removal. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface in fish as ammonia ions. Kidney do not play any significant role in its removal. Terrestrial adaptation is necessitated the production of lesser toxic nitrogenous waste like urea, uric acid for conservation of water. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians and marine fishes mainly excrete urea and are called ureotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, landcell, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures in most of invertebrates. These structures are simple tubular forms, whereas vertebrates have complex tubular organs called kidneys. Okay, some of these structures are mentioned here. Protonephridae or flame cells are the excretory structure in Platyhelminthes, means flatworm. For example, planaria. Rotifers, some annelids, and cephalochordate, means amphioxus. Protonephridae are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridae are the tubular excretory structure of earthworm and other annelids. Nephridae help to remove nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structure of most of the insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennate glands or gland glands perform the excretory function in crustaceans like prawns. So 16.1 Excretory system, human excretory system. In humans, excretory system consists of a pair of kidneys, one pair of ureters, urinary bladder, and urethra. Kidneys are reddish brown bean-shaped structures situated between the levels of large thoracic and third lumbar vertebrae, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in weight, 2 to 3 cm in thickness, with an average weight of 120 to 170 gram. Towards the center of the inner concave surface of kidney is a notch called hilum, through which ureter, blood vessels, and nerves enter. Inner to the hilum is a broad funnel-shaped space called renal pelvis, which projects called calyces. Inner to the hilum is a broad funnel-shaped space called the renal pelvis, with projection called calyces. The outer layer of kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calyces, singular calyx. The cortex extends in between the medullary pyramids as renal columns called columns of Bertini. Each kidney has nearly verminal complex tubular structure called nephrons, which are the functional units. Each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is tuft of capillaries formed by the afferent artery, a fine branch of renal artery. Blood from the glomerulus is carried away by efferent artery. The renal tubule begins with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus, along with Bowman's capsule, is called the Malpighian body or renal corpuscle. The tubule continues further to form a highly coiled network called proximal convoluted tubule. PCT. A hairpin shape Henle's loop is the next part of the tubule, which has a descending and ascending limb. The ascending limb continues as another highly coiled tubular region called distal convoluted tubule. The DCTs of many nephrons open into a straight tube called collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian corpuscle, PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called peritubular capillaries. Minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephron. 16.2 Urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion, that take place in different parts of the nephron. The first step in urine formation is the filtration of blood, which is carried out by the glomerulus and is found glomerular filtration. On an average, 1100 से 1200 ml of blood is filtered by the kidneys per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, that is the endothelium of glomerular blood vessel, the epithelium of Bowman's capsule, and the basement membrane between the two layers. The epithelial cells of Bowman capsule called podocytes are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slits for blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins pass on to the lumen of the Bowman's capsule. Therefore, it is considered as process of ultrafiltration. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins pass on to the lumen of the Bowman's capsule. Therefore, it is considered as process of ultrafiltration. The amount of filtrate formed by the kidneys per minute is called glomerular filtration rate, GFR. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liters per day. The amount of filtrate formed by the kidneys per minute is called GFR. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liters per day. The kidneys have built-in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus (JGA). JGA is a special sensitive region formed by the cellular modification in the distal convoluted tubule and afferent arteriole at the location of their contact. A fall in GFR A fall in GFR can activate the GG cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A fall in GFR can activate the GG cell to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A comparison of the volume of the filtrate formed per day 180 liters per day with that of the urine release 1.5 liters suggests that nearly 99% of the filter has to be reabsorbed by the renal tubules. This process is called reabsorption. The tubular epithelial cells in different segments of nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acids, sodium ion, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by the passive transport. The tubular epithelial cells in different segments of nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acids, sodium ion, etc. in the filter are reabsorbed actively, whereas nitrogenous waste are absorbed by the passive transfer. Reabsorption and water also occur passively in the initial segments of the nephron. During urine formation, the tubular cells secrete substances like hydrogen ion, potassium and ammonia. In the filtrate. During urine formation, the tubular cells secrete substances like hydrogen ion, potassium ion and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and acid-base balance of the body fluids. Function of the tubules. 16.3 Function of the tubules. Proximal convoluted tubule (PCT). PCT is lined by simple cuboidal brush border epithelium which increases the surface area for reabsorption. Nearly all the essential nutrients and 70 to 80% of electrolytes and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ions and ammonia into the filtrate and by absorption of bicarbonates from it. Les loop. Reabsorption is minimum in its ascending dip. However, this region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The descending limb of loop of Henle is permeable to water, but almost impermeable to electrolyte. This concentrates the filter as it moves down the ascending limb, is impermeable to water, but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to passage of electrolytes to the medullary fluid. Distal convoluted tubule (DCT). Conditional reabsorption of sodium and water takes place in this segment. DCT is also capable of reabsorption of bicarbonates and selective secretion of hydrogen and potassium ions and ammonia to maintain the pH and sodium-potassium balance in blood. Collecting duct. The long duct extends from the cortex of the kidney to the inner part of the medulla. Large amounts of water could be reabsorbed from this region to produce a concentrated urine. This segment allows passage of small amounts of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of hydrogen and potassium ions. 16.4 Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta play a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite direction and thus forms a counter current. The flow of blood through the two limbs of vasa recta is also in counter current pattern. The proximity between the Henle's loop and vasa recta as well as the counter current in them help in maintaining and increasing osmolarity towards the inner medullary interstitium, that is from 300 milliosmol in the cortex to about 1200 milliosmol in the inner medulla. This segment is mainly caused by NaCl, sodium chloride and urea. NaCl is transported by the ascending limb of Henle's loop, which is exchanged with the descending lip of vasa recta. NaCl is returned to the interstitium by the ascending portion of vasa recta. Similarly, small amount of urea enter the thin segment of ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. The above described transport of substances facilitated by special arrangement of Henle's loop and vasa recta is called counter current mechanism. The mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of sub-interstitial gradient helps in an easy passage of water from collecting tubule, thereby concentrating the filtrate, means urine. Human kidneys can produce urine nearly 4 times concentrated than the initial filtrate form. 16.5 Regulation of kidney function. The functioning of the kidneys is efficiently monitored and regulated by hormonal feedback mechanism involving the hypothalamus, GGA, and to a certain extent the heart. Osmoreceptors in the body are activated by changes to blood volume, body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate this receptor, which stimulate the hypothalamus to release antidiuretic hormone ADH or vasopressin from the neurohypophysis. ADH facilitates water reabsorption from latter parts of the tubule, thereby preventing diuresis. An increase in body fluid volume can switch off the osmoreceptor and suppress the ADH release to complete the feedback. ADH can also affect the kidney function by its constrictory effect on blood vessels. This causes an increase in blood pressure, means hypertension. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the GFR. The GGA plays a complex regulatory role. A foreign glomerular blood flow or glomerular blood pressure or glomerular filtration rate can activate the GG cells to release renin, which converts angiotensin in blood to angiotensin 1 and further to angiotensin 2. Angiotensin 2, being a powerful vasoconstrictor, increases the glomerular blood pressure and thereby GFR. Angiotensin 2 also activates the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of Na plus and water from the distal parts of the tubule. Aldosterone causes reabsorption of sodium and water from the distal parts of the tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as renin-angiotensin mechanism. Increase in blood flow to the artery of the heart can cause the release of arterial natriuretic factor ANF. ANF can cause vasodilation, dilation of blood vessels, and thereby decrease the blood pressure. ANF mechanism therefore acts as check on the renin-angiotensin mechanism. Micturition. Urine formed by the nephrons is ultimately carried to the urinary bladder, where it is stored via a voluntary signal given by the central nervous system. This signal is initiated by the stretching of the urinary bladder as it gets filled with urine. In response, the stretch receptor on the walls of the bladder sends signal to the CNS. The CNS passes on motor messages to initiate the contraction of smooth muscle of the bladder and simultaneous relaxation of the urethral sphincter, causing the release of urine. The process of release of urine is called micturition, and the neural mechanism causing it is called micturition reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. Urine form is a light yellow colored watery fluid which is slightly acidic, pH 6, and has characteristic odor. On an average, 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristics of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidneys. For example, presence of glucose, glycosuria and ketone bodies, ketonuria in urine are indicative of diabetes mellitus. 16.7 Role of organs in excretion. Role of other organs in excretion. Other than the kidneys, lungs, liver and skin also help in elimination of excretory waste. Our lungs remove large amount of carbon dioxide, approximately 200 ml per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secretes bile containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins and drugs. Most of these substances ultimately pass out along with digestive waste. The sweat and sebaceous glands in the skin can eliminate certain substances through their secretions. Sweat produced by the sweat glands is watery fluid containing NaCl, small amounts of urea, lactic acid, etc. Though the primary function of sweat is to facilitate a cooling effect on the body surface, it also helps in the removal of some of the wastes mentioned above. Sebaceous gland eliminates certain substances like sterols, hydrocarbons and waxes through sebum. This secretion provides a protective oily covering for the skin. Do you know that small amounts of nitrogenous waste could be eliminated through saliva too? Disorders of the excretory system. Malfunctioning of kidneys can lead to accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In some patients, urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from the convenient arteries pumped into a dialysing unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialysing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid, dialysing fluid, having the same composition as the plasma except the nitrogenous waste. The porous cellophane membrane of the tube allows the passage of molecules based on concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The clear blood is pumped back to the body through a vein after adding antiheparin to it. This matter is to be boon for thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in correction of acute renal failure, kidney failure. Functioning kidney is used in transplantation from a donor, preferably a close relative, to minimize its chances of rejection by the immune system of the host. Modern clinical procedures have increased the success rate of such complicated technique. Renal calculi. Stone or insoluble mass of crystallized salts, oxalates, etc. formed within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney.